I would like to welcome you to the first laboratory in TBIO 572, the pathologic basis of cancer. The learning objectives of this session are twofold. First, by the end of this session, you should know the function of the various components of the lights microscope and how to optimize the resolution by color illumination. For this, I refer you to the 3 videos posted on YouTube entitled Intro to the Lights Microscope. And second, you should become familiar with the basic concepts of histology. Now, I realize that most of you have not taken a class in histology as undergraduates. So in the present video, I'm going to try to give you a brief summary of histology. Now, typically a course in histology starts out by teaching the basic tissues before proceeding on to the organ systems. In histology, the term tissue has a specific meaning. A tissue is defined as a collection of similar cells and their associated extracellular matrix. There are 4 basic tissue types that should be learned. These are epithelium, connective tissue, nerve, and muscle. Of course, each of these basic tissues can be further subdivided, as I will describe in a minute. After learning the basic tissue types, the student then goes on to study the organ systems, which are subdivisions of the body, such as the liver and the skin. Each organ, in turn, is made up of the 4 basic tissue types. Now, as you go through the slides in this laboratory, I ask that you classify the various structures you observe into one of the 4 basic tissue types. As I indicated before, each of the 4 basic tissue types can be further subdivided as illustrated in this very simplified chart. For example, epithelium can be subdivided into simple, which means that it is only one cell layer thick, stratified, which means that it is multi-layered, and pseudostratified, in which some, but not all of the cells, reach from the base to the free surface. Next comes the connective tissue, which is a catch-all category. It includes collagen, fat or adipose tissue, cartilage, and bone. In addition, blood, which is not listed here, is also classified as connective tissue. After that is nerve tissue, which consists of neurons that can actively transmit signals, and glial cells that are the support cells. An example of a glial cell is a Schwann cell that wraps myelin around the axons. And finally, there is muscle tissue, of which there are 3 types: skeletal, cardiac, and smooth. Now, when you start out, there is a natural temptation to put the slide on the microscope and go directly to the 40x objective lens. Resist this temptation. I want you to go through the slides in the following fashion. First, place the slide on a white sheet of paper, as shown here, and just look at it. Determine the general layout of the tissue, the number of pieces, and the nature of the staining. Next, place the slide on the stage of the microscope and examine it under the 4x objective lens. Remember that the extra condenser lens must be rotated out of the way as shown here in the left panel. After scanning the slide with the 4x objective, switch to the 10x objective lens. Again, remember to rotate the extra condenser lens back into position as shown on the right panel. I recommend that you examine the periphery of the tissue first and then the center. Finally, after you locate something interesting, switch to the 40x objective lens. In general, the focus at 40x should be close to that of the 10x objective lens. However, you may need to adjust the condenser diaphragm. Start with slide 53 of a normal non-secretory mammary gland. By eye, note that the sample consists of a single piece with a light pink or eosinophilic matrix, in which are embedded small dark pink regions. Under the 10x objective lens, note that the light eosinophilic staining region is composed of adipose connective tissue. This consists of holes in the section where the fat used to be. Most of the fat had been extracted by solvents used during processing, leaving only holes surrounded by a thin rim of cytoplasm. The darker eosinophilic region is composed of collagen, another type of connective tissue. And finally, you will also see blue or basophilic structures within the collagen that belong to the ducts and glands, and of course these are classified as epithelial tissue. Now switch to the 40x objective lens. At this magnification, you can observe that the collagenous connective tissue is eosinophilic, wavy, and contains few cells. Interspersed within the collagen are the fibroblasts that have produced the collagen. These are indicated by the nuclei of the fibroblast. Also note that most of the ducts and the glands are located within the connective tissue and lined by epithelium. Next, I want you to look at slide 55 of a lactating mammary gland. Even by eye, it is apparent that this section is quite different from the previous non-lactating specimen. For one thing, there is less of the light-staining adipose tissue. And secondly, it has a more blue or basophilic character to it. This means that it contains more cells, since it is the nuclei of these cells that stain blue with hematoxylin. Under a low magnification, locate one of the secretory clusters. The lumen of these glands are large and contain secretory material. This portion is lined by simple cuboidal epithelium, while the larger ducts are lined by stratified cuboidal epithelium. Also, identify blood vessels by their content of erythrocytes, which are very eosinophilic. Under the 40x objective lens, observe that the epithelium lining the glands are simple, that is, one layer of cells thick. In the blood vessels, you will observe the individual erythrocytes, and remember that they are approximately 7 microns in diameter. The arteries and the veins often occur in pairs as shown here. Also note that the blood vessels are surrounded by a layer of smooth muscle that you can recognize by the fact that it stains a dark pink and that the nuclei are spindle-shaped. Typically, the layer of smooth muscle surrounding the artery is thicker than that surrounding the vein. Now examine slide 97 of the monkey lungs by eye. Observe that this section is light staining because most of it consists of empty space, where the air was. Also, be advised that this slide is old and the blue hematoxylin has faded in places. Observe this section under the 10x objective lens. As shown on the right half of this slide, most of the lung consists of a meshwork of epithelium where gas exchange takes place. This represents several layers of simple squamous epithelium closely opposed to each other. On the left side of the slide is a bronchus, which is a large air passageway. It is surrounded by a layer of collagenous connective tissue that also contains cartilage as well as smooth muscle. This figure shows a higher magnification view of the wall of the bronchus. The epithelium of the bronchus is lined by pseudostratified columnar epithelium. Remember that pseudostratified means that most, but not all of the cells extend from the base to the free surface. On the free surface, you may be able to distinguish hair-like structures that are the cilia. In the wall of the bronchus is a band of smooth muscle as well as cartilage. The cartilage typically has a glassy appearance with round chondrocytes scattered throughout. Next, examine slide number 126 of the normal human prostate. This section consists of a single piece with an eosinophilic capsule on the outside and a basophilic glandular region on the inside. This is a low magnification view of the junction between the capsule and the glandular region. Note that the capsule consists of light and dark eosinophilic regions. The light staining region is collagenous connective tissue, while the darker regions are bands of smooth muscle. Another way to distinguish between these two tissues is that the smooth muscle contains more nuclei and they tend to be more spindle-shaped. Of course, the glandular region on the lower right stains basophilic. At higher magnification, you can observe that the secretory portion of the glands are lined by cuboidal to columnar epithelium. Note that most of the nuclei are arranged in a row. Now examine slide number 138 of the human cervix by eye and observe that it consists of one piece that is shaped like an arrowhead. Although it is hard to tell without magnification, the nature of the epithelium changes near the point of the arrow. When one surface is viewed under the 10x objective lens, the epithelium is found to be simple columnar. That is, it is one cell layer thick, and the epithelial cells have a columnar shape. In contrast, the epithelium on the other side of the section is quite different. It is classified as a stratified, squamous, non-keratinizing epithelium. The term stratified means that it is multilayered. The term squamous means that the cells on the free surface have a flat shape, and the term non-keratinizing means that the epithelial cells retain their nuclei all the way to the free surface. This is a high magnification view of an anterior region of the section, which contains a mixture of smooth muscle and collagenous connective tissue. Again, as compared to the collagenous connective tissue, the smooth muscle is darker staining and the nuclei are more elongated. The final slide I ask you to examine is slide 37 of a lymph node. Without any magnification, you can see that the slide contains a single piece and is relatively basophilic or blue. This suggests that it has a high cellular content, since most of the basophilia is due to the nuclei of cells. When you examine this slide under low power, you will observe that one side of the node is surrounded by adipose connective tissue, which is easy to identify because of the many holes. The node itself is surrounded by a thin capsule of collagenous connective tissue. The body of the node is composed of a multitude of lymphocytes. In one area, termed a germinal center, the lymphocytes are undergoing the process of activation. At higher magnification, you can better appreciate that the lymphocytes are small cells that consist of simply a nucleus that is surrounded by a thin layer of cytoplasm. Each of the blue dots here is the nucleus of a lymphocyte. In the germinal center, the activated lymphocytes have expanded their cytoplasm, which makes this region stain slightly lighter than the rest. You may wish to examine this slide under 100x oil immersion. For this, I refer you to the video on oil immersion microscopy.